Hey, welcome back, biology honor students. In this episode, we're going to be breaking down all things DNA. So let's not waste any of your time. Let's get right into it.
It's hard to breathe, but that's alright. Hush.
DNA, as you know, location, nucleus, that should be something we're very familiar with. And the function of DNA. DNA controls all things traits. I mean, you're looking at me, so we can talk about some traits. Size of the nose, eye color, hair texture, size of the ears, everything about what can be physically seen, height, bone structure, bone density, anything that can be physically seen as far as a organism, human, whatever, animal, that would be considered a trait. So everything, all those decisions are made by the DNA. Now, when we talk DNA, we certainly want to understand structure. And you can see here the diagram just immediately beside me there in the white background. It's a kind of a breakdown of the chemical structure of DNA, and in the gray ovals you can see the names, but we've got a phosphate group, we've got a sugar group, and we've got a nitrogen base group, and those are those letters A, T, C, and G that hopefully you're familiar with. But that phosphate group, super important. And the sugar is the one that creates the most variation. We're talking DNA, so we're talking deoxyribose. And later on, when we're talking RNA, we'll just be talking about ribose as far as that sugar. And in this case, in this DNA, our nitrogen bases, A, T, C, and G. And of course, again, that will change once we are talking about ribose. Okay, so when we're talking DNA, we're going to go through the process of replication, transcription, translations, all with the idea of giving you a good solid base for how we get those proteins. And in your modules this week, you're going to be going through the DNA steps and the variations of everything. Some of it from a genetics standpoint, other from a structure standpoint. So we're going to cover all bases to try to get you as much ammunition as you can. But as you can see here, we've got replication. And replication simply refers to the copying of the DNA strands. And we would first want to emphasize in that far diagram to the far side over there, the green and the blue with the two strands. Hopefully you're familiar with this process. But if not, it simply goes, the DNA strand is opened up. You know, it's a double-stranded helix, and so they're separated, and then each strand is in red. A polymerase will run down there, and if it encounters an A, then it'll put a T on the new strand. If it runs across a T, then it'll put an A in the new strand. And a C, it'll put a G, and in a G, it'll put a C, keeping those complementary base pairs. And in that oval at the bottom left-hand corner, you can see there the upper row of letters is the old strand that is being read, and the bottom is the new strand that is being read. And you can see that if polymerase sees a T, it'll put an A, and if an A, it puts a T, and then a G and a C, and on through there, and that creates that new strand. But the cool factor of this, and it's there on the screen now, that once that is done and they reconnect those strands, what you have is an older strand is paired with a newer strand. And what that does for the DNA, that ensures that there's always, it keeps it young, keeps it as fresh as possible. The idea that half of it is always a brand new strand, and then what's considered the older isn't really that old, and so it keeps everything as fresh and efficient and as young as possible. Okay, now what we're going to look at is transcriptions. Okay, transcription is the idea of now we've duplicated that DNA strand, and now what we're going to do is convert it to an RNA strand. And in that graphic up at the very top with the pinkish circle, and I know you're thinking, well, that's not big enough really to see, and it's not that you need to see all the little letters and the words and stuff, just the overall concept of what's happening. You can see that the DNA, again, has been opened up. Okay, and then that red strand hanging off there, that's the new RNA strand. So the same thing that happened during replication happens again, except this time it's making an RNA strand. So again, that polymerase will get on there and begin to read, and the change now is, and you can see it in the upper right hand corner, that gray oval, that if it comes across, or it needs to put a T in there, it now puts in a U, which makes it into an RNA strand. And then the sugar is then converted to a ribose to make the RNA strand. So you'll have, again, the lead nucleotides being read, and if it comes across an A and it's supposed to put a T, it will put a U instead. But everything else remains the same. If it comes across a T, then it'll put an A there. If it comes across a C, it'll put a G there. If it comes across a G, then it'll put a C there. So those other three remain the same. The one change is now that we've got, if we're supposed to put a T, we put a U. Then, for the next step, what goes on is they're kind of grouped in groups of three, and that's called a codon. Groups of three, and on the next slide, or next screen, as you can see there, those codons are going to be read, and that determines the amino acid. But that's for next time. So here we are. Now we're at translation. Okay, translation, just like the word mentions, is that we are changing it. No, we are now making the proteins. So again, you can see here, again, the diagram, you may be thinking that's pretty small, but it's not that you need to see all the words. We're just going to talk about what's going on. But that big kind of greenish blob in the diagram, kind of is in the lower left-hand corner, but it's in the center of all the figures there, symbols there. That's a ribosome. Okay, and a ribosome is super important. One of the organelles that hopefully you're familiar with. But the ribosome will basically attach itself to an RNA strand, and the RNA strand kind of runs through it, just like an old film projector, or just something that runs through, and ribosomes kind of wrapped around it, and it's reading it. And those yellowish kind of cross symbols or figures, those are tRNA. And in the tRNA, we'll add the appropriate nucleotides to form the anticodon. We've got the codon that's being read, those groups of three, and in the tRNA will match up the appropriate nucleotides. In other words, if it's an A, it'll put a U. If it's a C, it'll put a G. And if it's a G, it'll put a C there. And if it's a U, it'll put an A there. But they do it in groups of threes. And the codon is on the RNA strand, and what the tRNA is adding is called the anticodon, or the opposite of what is going on. And so once those anticodons are added, you can see then the little row of circles at the top of that diagram. There's a blue and a yellow. Then it looks like a light purple, then maybe a teal, then an orange, and a pink, then a gray. Those are representing of amino acids. So whatever the anticodon in other words, what collection of three nucleotides it is, determines what amino acid is added to the chain. And that chain just kind of runs along or off of the top of right above the ribosome. And if you look at the gray square in the far upper right hand corner, you can see again that represents the anticodons being formed, and then stringing from that the appropriate amino acids. And of course, then whatever the collection of amino acids are, that determines what the protein is. So, and what that means is that the string of amino acids is evaluated, and whatever the order is creates a particular protein to do a particular job. So you got your ribosome, which is that big green blob that reads the RNA. The tRNA adds the appropriate anticodon, or the collection of 3 nucleotides based on the RNA rules. Those form the amino acids, which eventually form the protein. Well, hopefully that helps you to speak as you go through your modules, having this information about DNA, RNA, but it's a whole idea of you start off with DNA, that gets converted, you duplicate that gets converted to RNA in translation, and then in transcription into the amino acid chain, which forms the protein. So hopefully that helps. If you need more information, hey, as always, you can always contact me, shoot me a canvas, get a hold of me somehow, and we can talk more about these processes. But until Wednesday, when we have our midweek check-in, we will see you later.